What came first, the chicken or the egg? It's a classic paradox. You can't get a chicken without an egg, and you can't get a chicken's egg without a chicken. In the world of biology, at the very origin of life, scientists faced an almost identical and far more profound chicken and egg problem. It's called the DNA-protein paradox. Think of it like this. DNA is the master blueprint. It contains all the instructions, all the code needed to build and run a living cell. But on its own, DNA is just a library of books that no one can read. To read the code and build the machines, you need proteins. Proteins are the workers, the enzymes, the molecular machines that do everything, including the vital job of copying the DNA. So you have a perfect catch-22. To get proteins, you need the instructions from DNA. But to read and copy the DNA, you need proteins. Which came first, the blueprint or the builder, the software or the hardware to run it? For decades, this was a brick wall for the theory of evolution. A spontaneous origin of life seemed impossible if you needed two incredibly complex, interdependent systems to appear at the exact same time by pure chance. But then a hero arrived, a clever idea that promised to solve the paradox. It was called the RNA World Hypothesis. The idea was brilliant in its simplicity. What if, before DNA and proteins, there was a single molecule that could do both jobs? A molecule that could both store information, like DNA, and act as a worker machine, like a protein. That molecule was RNA. The story goes that early life was an RNA world. RNA molecules stored the genetic code, and other RNA molecules, called ribosomes, catalyzed the reactions needed for life to begin. It was the perfect solution. It was the chicken and egg all rolled into one magical molecule. This story is now the dominant theory for the origin of life. It's presented as the most plausible scientific pathway from non-life to life. But today, we're going to look closer, and we're going to see that the RNA world isn't a solution to the origin of life. It's an admission of defeat. It's a story that, under the slightest scrutiny, completely refutes itself. Problem number 1. The miracle before the story begins. The RNA World Hypothesis starts with a huge assumption that the primordial soup was filled with a rich supply of ready-to-use RNA molecules. But where did they come from? An RNA molecule is itself an incredibly complex chain built from smaller units called nucleotides. Each nucleotide has 3 parts: a phosphate, a sugar called ribose, and a base. Getting these components to form and link up by chance in a primordial soup is a chemical nightmare. The sugar, ribose, is notoriously unstable. It breaks down in water. Geochemists have called the idea of a natural supply of ribose a fantasy. Even if you somehow got the sugar, attaching the base and the phosphate in the right way without destroying the whole structure is another near impossible chemical hurdle. In simple terms, to get the RNA world started, you need a miracle of chemistry that is arguably even more improbable than the original DNA-protein problem it was trying to solve. It's the story refutes itself before it even starts. Problem number 2. A world made of sandcastles in a hurricane. Let's be generous. Let's imagine that by some magic, a perfect RNA molecule appears. The story requires this molecule to survive for a very long time, to replicate and evolve. There's just one problem. RNA is an incredibly fragile and unstable molecule. It is so unstable that it breaks down rapidly, especially in water and when exposed to minerals or UV light, all the things that were supposedly abundant on the early Earth. Biologists today have to store RNA samples in ultra-cold freezers under controlled conditions to stop them from degrading in minutes. The idea of an RNA world is a misnomer. It wouldn't have been a world, it would have been an RNA moment, a fleeting, fragile existence. It's like trying to build a sandcastle in the middle of a hurricane. The environment that supposedly formed it is also perfectly designed to destroy it. Problem number 3. The myth of the supermolecule. The whole hypothesis rests on the idea that RNA can be a jack of all trades. It can store code, and it can do work. But how good is it at these jobs? Compared to DNA, RNA is a terrible information storage molecule. It's unstable and prone to errors. Compared to proteins, RNA is an incredibly clumsy and inefficient worker. The catalytic abilities of the best lab-engineered ribosomes are pathetic compared to a standard protein enzyme. And the biggest secret? The ability for an RNA molecule to self-replicate, the one function this whole story absolutely depends on, has never, ever been observed in nature. Scientists have spent decades trying to synthesize a self-replicating RNA molecule in the lab and have failed spectacularly. So, the story asks us to believe that a molecule that is terrible at both jobs and which possesses a mythical, never-seen primary function was the foundation of all life. Problem number 4. The final act of self-destruction, the impossible transition. This is the most devastating, self-refuting flaw of all. Let's imagine the impossible. Let's say the RNA world existed. It was stable. It had self-replicating RNA that could do all the jobs needed for a primitive cell. Now what? How and why would this perfectly functional RNA world decide to invent a completely new, far more complex system based on DNA and proteins? And then, how would it transfer all its information to this new system and then make itself obsolete? Think about it. This is like a fleet of propeller airplanes deciding to invent the jet engine, designing a system they have no concept of, building the first jet while still flying, and then gracefully retiring as soon as the first jet takes off. It's a logistical and logical absurdity. There is no step-by-step evolutionary pathway from an RNA-based system to a DNA and protein-based system. The RNA World Hypothesis isn't a bridge from non-life to life. It's a fantasy island with no bridges connecting it to the chemical mainland on one side and no bridges connecting it to the world of real biology on the other. It was invented to solve a chicken and egg paradox, but it ended up creating 3 more. It's the ultimate scientific ghost story. A story that sounds good in the dark, but vanishes the moment you shine the bright light of chemistry and logic upon it.